enabling proteins for synthetic biology and bioanalytics.
Hi everyone, welcome to RNAct webinars, a series of online seminars on biology adjacent topics. Every seminar aims to give an overview of the topic and can be used as a learning material for study, depending on the topic for high school, for bachelor, master, or even students starting their PhDs. My name is Anna. I'm here with my co-host Luca. We are PhD students within the RNAct project, funded by European Commission, a part of the Marie Skłodowska-Curie Action Innovative Training Network. Today we are talking about the beautiful complexity of RRM-RNA interactions. Here is a typical digital visualization of these molecules. They can interact with each other and form a complex. These two molecules and their complexes are crucial for our bodies. They take a big part in keeping us alive. Our objective for today is to give you a general idea about these two molecules and their complex. What are they, what are their properties, and why studying them is important. We will begin with RNA molecule, and we will talk about what is it, what is RNA structure, and how RNA can interact with proteins, including RNAs. RNA stands for ribonucleic acid. This molecule is a polymer, which means that it consists of many similar building blocks connected together. Such blocks of RNA is called nucleotides. On the atomic level, nucleotide is a set of oxygen, hydrogen, carbon, phosphate, and nitrogen atoms. They are held together by the chemical bond in this specific pattern. The following subunits are recognized: the phosphate group, the ribose, and this one is also called sugar. And also this is the way in which RNA molecules differ from DNA molecules, because RNA molecules has ribose in its nucleotides, when DNA has deoxyribose. And the last subunit is nitrogen base. Here is a 3D visualization of a nucleotide along with its subunits. The sugar and the ribose of a phosphate group are, generally speaking, constant subunits, and they do not change. But the nitrogen bases can be different for different nucleotides. There are 4 canonical nucleotide bases: adenine, guanine, uracil, and cytosine. Each has a slightly different chemical formula. And this is the visualization. The blue color stands for nitrogen atoms, the pink for oxygen, and green and red for the carbon atoms. Bases are classified as purines and pyrimidines. Purines consist of 2 rings and pyrimidines just 1. 2 or more nucleotides can form a chain by bonding with each other via phosphodiester bonds between the sugar of the first nucleotide and the phosphate of the second one. This line of sugars and phosphates is known as the backbone of RNA, and the basis is known as a side chain. Here is how it looks in 3D. Synthesis or creation of RNAs in eukaryotes, aka organisms whose cells have a nucleus, it is happening directly in the nucleus. The central dogma of molecular biology stands that DNA makes RNA makes protein. And you can find out more about how RNAs and proteins are made in the online seminar by Anna Perez, another PhD student from RNAct project. Now, let's talk about structure and why it is extremely important. In the world of molecules like DNA and RNA and proteins, the structure of a molecule defines its function, defines what it can and cannot do. Think about key analog. It's a pretty close analogy. Some RNAs cannot bind given protein at all. They simply don't fit, the shape not corresponding. Some RNAs are not exact fit, but it can work for a bit, maybe for a really small amount of time. The binding is really weak. And finally, some RNAs do bind given protein strongly. And in this case, they form a complex which can perform some functions within the cell. An example of such process is splicing. Some RNA molecules are designed to transfer genetic information from the nucleus to the cell itself. But upon synthesis, they contain regions with genetic information, here shown in green, and without it, in the orange. And the region without genetic information have to be removed from RNA before the whole process can begin. The remaining parts of RNA with genetic information are joined together. And this process is done by 5 protein RNA complexes, exactly those which we've been talking about before. Let's come back to the structure, which defines functions. There are 4 levels of RNA structure: primary, secondary, tertiary, and quaternary. Primary structure is a sequence. The sequence is a string of bases of the RNA written from 5' to 3', where 5' marks nucleotide which phosphate group is not binding to any other nucleotide. And here is the example. This chain has uracil, cytosine, and guanine, and its sequence is going to be UCG. RNAs differ in length. Some are really short, for example, shorter than 100 nucleotides, but some can be pretty long, for example, a few thousand nucleotides. Primary structure on its own does not really give a clear vision of the structure, but it's a foundation of it. Secondary structure is the first step towards describing how RNA molecules can fold. It is all about describing the base pairing. Base pairings are hydrogen bonds that connect 2 bases of 2 different nucleotides. Normally, cytosine pairs with guanine, and adenine pairs with uracil. The base pairing is the main way for RNA to fold on itself. It means that if, for example, we have this RNA sequence, then its secondary structure may look something like this. Some of its nucleotides are paired, and some are unpaired. The unpaired part is called single-stranded, and the paired part double-stranded. Here's a little memo to remember canonical base pairing. Car in the garage for C pairs with G, and alien in the UFO for A pairs with U. You should be aware that non-canonical base pairing, for example AG, can occur and actually are pretty common, and this is the way how RNA can fold in the vast range of structures. Let me see some examples. Often RNA fold into a helix like so, and this element is called loop. Together they can form a structure which is called hairpin. And basic elements can form quite curious structures. Feel free to pause the video to take a closer look. Longer RNAs can form more intricate structures like so. But is this schema is actually enough to describe the folding? Well, not really. For example, in reality, there is a connection between these two residues, these 2 nucleotides, but there is actually no way to show it easily on this 2D schema. So we need to move on to the 3D. Tertiary structure is exactly this. It's a digital 3D image of a molecule. So this is how such a secondary structure looks in 3D. This representation is called sticks. It's really good for looking on the nucleotides up close, but it can be a bit confusing if we zoom out like this. It is easier to see overall folding using a cartoon representation. For example, here the position of the backbone is much more clear than on the previous image. And also it is possible to combine both of those representations. Sometimes it's really useful to color nucleotides according to which part of the structure they correspond, like so. Longer RNAs can fold into more intricate structures. On the left, you can see a part of the cricket paralysis virus, and on the right, this is the RNA which form a complex with the RRM, and they both take part into the splicing process, which we talked about before. Last but not least is quaternary structure. It depicts 2 or more molecules bounded together. Here you can see a complex formed by 1 single-stranded RNA chain and 3 protein chains. Its PDB ID is 3BOY. PDB stands for Protein Data Bank, and this name identifies this specific complex out of all known to humanity structures. And here we have another one. As you can see here, there are 3 RNA present, and 2 of them are having more complicated structures than the RNA from the previous one, because here we have both single-stranded parts and double-stranded ones. But how those 2 molecules can interact with each other? Well, mostly it's happening through the electrostatic interactions. The 3 types of those are hydrogen bonds. Those are medium strengths, van der Waals interactions, the low strengths, and salt bridges, they are really high strengths. Here you can see an example of RNA base binding with a protein through 2 of those hydrogen bonds. And here the molecule of water forms hydrogen bond with sugar of the RNA and with a part of the protein. So those two are connected to each other. And here an example of RNA backbone having again a hydrogen bond with the protein. Another type of the interaction are hydrophobic interactions and stacking. Here you can see how the ring of RNA is stacking with the ring of the protein. This brings us to the last slide for the RNA part of this webinar. Here you can see a complex of RNAs and RNA molecules. And from here onwards, Luca is taking over.
Thank you, Anna. Hi, I'm Luca, and in this second part of the webinar, I will give you a small overview on proteins, how they are built, and what are some key features to then go more into details about protein and RNA interaction and when we want to study them. To start, it's good to define what a protein is and how it is made. So when we talk about proteins, we are referring to a class of biomolecules built from the same building blocks in a similar way as you saw for the RNA. In case of the proteins, these building blocks are the amino acids. All the amino acids that we can find in our proteins usually have a constant part, highlighted in gray, and a variable part that highlighted here in orange. This part is usually called as side chains, and it what gives the flavor, let's say, to the different amino acids. So it's really important for the formation of the final protein. In our body, we usually find 20 different flavors of these amino acids, everyone differing from each other, as you can see, from the different side chains that they have. And so basically all having different properties respect to each other. And so giving also at the end different properties to the proteins that are part of. So these monomers they can actually bind to each other through their extremities of their constant part, forming basically a chain of different amino acids in a sequence. So this is actually what we refer as primary sequence of a protein. So with primary sequence, we usually refer at the sequence of amino acids that are forming the protein, in a similar way as you saw with the nucleotides for the RNA. Then these chains in the environment of the cell can adopt different shapes in the 3D space, creating some structural elements. Some of these are really common and recurring in proteins and many times, and it's what we refer as secondary structure elements. Two of the most important and most common one are the ones that are shown here. So we call it as beta sheet or alpha helix, depending basically on the shape they adopt in the 3D space. And obviously then we can combine these different structural elements to form what at the end is we refer as tertiary structure. In here we can find the different secondary structure elements together and combined together with some less structure part that we usually call loops, and is at the end this tertiary structure that gives protein its future and its function. So it's based on this final shape that the protein can carry its function inside the cell. So, for example, digesting other molecules, function as a storage for example, ions, or as we will see briefly, binds RNA molecules. But before to go on, it's useful to define one last concept. When we look at the proteins many times, we find both more structured part and more unstructured part that can play different roles. Usually we refer as structured part as domains. We define as domain that part of a protein that can fold properly and carry its function independently from the rest of the protein. So in a protein, many times what we find is many different domains that can have either similar function or different function. And altogether they are important for the final function of the protein inside the cell. Among these different domains, the one we are interested in and I will talk about is actually what we call RNA recognition motifs. This is a subset of a bigger family of the RNA binding domains that, as the name suggests, their function is to bind and recognize RNA. These RNA recognition motifs domains are usually consisting of 90 to 100 amino acids, and what we find usually is in the proteins is not only one of these domains, but many times we find more than one, for 2 or 3 different RNA recognition motifs. These RNA recognition motifs are actually quite abundant among the RNA binding domain family, and we can find them in many different key processes of the cell cycle and cell life. So the malfunction of these small domains can cause severe diseases in the organism and can cause, for example, cancer or spinal muscular atrophy. So in the example I'm showing here, we can identify 2 of these RNA recognition motifs, RRMs for short, and looking at the structure of these RRMs domains, we can identify some recurrent structure elements. For example, in the center of this domain, we find a series of amino acids forming what we saw before is called beta sheets. And these beta sheets are actually a key feature in the RRM structure, but also in the RRM's function. This surface is in fact the platform with which the RRMs form the important interaction for binding the RNA, where there are few amino acids that can interact and acts as anchor for the RNA-RRM interaction. Then we also have these other additional parts of the proteins that are the alpha helices or the loops, as I mentioned before, that can also play a role in helping this beta sheet to recognize the RNA molecule, and altogether play a role in the binding and recognition of the specific RNA sequences. So this recognition of the RNA is happening through different interaction of different natures that are exactly the same that Anna already introduced previously when she introduced the RNA. And some of them are helping for the strength of the binding, but other are useful for the specific recognition of our RNA sequence respect to the other. And through this platform, one single RRM is able to recognize 3 to 4 nucleotides of the RNA molecules. So having 2 of them, the length of the basis of the RNA that the protein can recognize, it's increasing. So we can, with 2 of these RRMs, we can basically recognize longer RNA sequences and also with more specificity. But when we are studying this interaction, we don't only have to consider the protein side, but also we have to remember that the RNA can adopt different secondary and tertiary structure. So also the RNA has a certain shape in the space, and also this is really important when we try to understand which are the molecular factors that are important for the binding and the recognition between the RRM domains and the RNA molecule. So the question now is why we want to study and why we are focusing on this specific interaction between the RRMs and the RNA. Well, our goal in the RNAct project is try to crack the code that allows one RRM domain to recognize a specific sequence of RNA. In particular, we are focusing on RRM domain as there is a big amount of information and experimental data to start with that we can use to investigate from a molecular level what is happening to try to understand the code underneath. So to also visualize it in a different way, we can think of an RRM as a molecule that have different binding site that can recognize specific residues of the RNA thanks to the different shape and different interaction. So basically, one RRM will bind a specific RNA. But now let's say that we know exactly how this is happening. So we know the code and we want to modify our RRMs to bind a different RNA molecule. So in this case, if we try to modify the RRM, we try to modify it in such a way that it binds not anymore the previous RNA molecules, but to now bind a different RNA molecules. So try changing the specificity of one RRM for this RNA. And this is really important as I mentioned before, these RRMs are involved in many key steps of the cell cycle and biological processes. And the mutation of just some residues, both on the RNA side or the RRM side, can cause severe diseases. For example, as I mentioned before, cancer, but also it can cause spinal muscular atrophy. So we can go back to the splicing process that Anna described before. And just to recap quickly, what's happening here is that our RNA that we need for the production of proteins has to be cut in the right way, dividing what is needed actually for producing our protein and some not useful parts of the RNA. So there are some proteins containing usually RRMs that recognize these regions that are not useful, and they are recognizing them and basically cutting them out to form the final RNA that is actually the one that it will go and produce the healthy, let's say, protein. And so there's no problem and the biological processes are all going fine. But for example, in the spinal muscular atrophy that I mentioned before, what's happening is that at the level of the RNA, there is just one mutation, and this impair the recognition of these proteins of the right regions that have to be cut out. So this time, these proteins cannot recognize properly the regions that have to be cut out. So what's happening is that also a small part of the useful RNA sequence that we need for the production of the protein is cut out, forming a different RNA molecule that results in a modified protein that is not working anymore, and then causing disease. So if we can know exactly how the RRM can recognize a specific sequence, as I told you before, we can try to modify one of these proteins that are actually recognizing the sites for cutting the RNA. So in this case, if we modify to recognize this time properly also the mutation, what's happening is that then the splicing process can happen again in a correct way. So in this time we modify the RRMs, and then it's able again to recognize the proper sequence to then cut out these regions that we don't need, and so restoring the actual proper RNA that can then result again in the correct production of the active protein. So in principle, allowing the cell to produce the healthy protein, and so trying to cure this disease. So hopefully I gave you a nice idea of why this is so interesting and useful to study this RRM-RNA interaction. And with this, I'm at the end also of my part. Hope you enjoy this webinar.
RNAct. To learn more about this project and its progress, you can follow us on several social media channels, with all the relevant information available at www.rnact.eu.